Morning, everybody. Welcome back to the 2022 COVI lecture series. My name is Dan Dakin, and once again, I'll be your moderator and host for today. As usual, our lecture today will run about 45 minutes, and then there'll be the opportunity to ask some questions at the end. So please feel free to put any questions into the chat box that you can find just beside the screen, the video screen here, and I'll pass those along once we get to the end. So today, I'm very pleased to welcome Sudarsana Poojari as our speaker this afternoon. Sudarsana is a senior scientist at the Cool Climate Enology and Viticulture Institute right here at Brock University. Sud received his PhD in plant pathology from Washington State University. And before joining COVI, he completed his NSERC postdoctoral fellowship at Agriculture and Agri-Food Canada, which is in Summerlin, BC. At COVI, Sud leads multiple research projects focused on grapevine virus epidemiology, advanced molecular diagnostics, and disease management aspects. And today, Sud is going to share a lecture titled Emerging Virus Diseases of Grapevine, Advances in Diagnosis and Management. So with that, I'll say welcome, Sud. I will turn it over to you. Please share your screen, and thank you in advance.
Thank you, Dan. Just trying to share the screen here. Can you see?
Yep. Yeah, looks good there.
All right. So push the presentation button here. Again, thanks. It's always nice to be part of COVI lecture series. Today, I'm going to talk about the virus diseases of grapevines, as Dan mentioned. By definition, let's start with the definition of what is the emerging grapevine virus diseases. Emerging viruses means any viruses or virus diseases that are newly discovered or having the potential to mutate and increase its incidence in the real world. So that being said, in that definition, many viruses fall in that definition, including the COVID-19 that we all know about and its variants. So, but we are not talking about COVID-19 today. We'll talk about the viruses that infect grapevines. The outline of my presentation here, as mentioned, we're going to talk about the importance, especially how we can identify or take some clues of this symptomology of the grapevine virus diseases, as well as how to detect them, both biological and molecular ways of detections, and the management aspects, especially in terms of the clean plant program or any insect vectors that can spread these virus diseases and how to manage those ones. So the first, the importance of the grapevine virus diseases. Before going to the major grapevine virus diseases and causal agents of those diseases, I just want to bring this to your attention that most of the grapevine production in North America, especially here in Canada, we grow grapevines along with palm fruits or stone fruits. I mean, especially apples and peaches. In terms of stone fruits, we have cherries, plums, apricots, and nectarines. You might see these orchards as well as vineyards side by side in many occasions. So what that brings to us as a virologist here is we are looking at if there is any viruses that cross-infect among these fruit trees. If you look into the literature and whatever the viruses, what we know so far, or characterized so far, this is unique. As you can see, there are so many viruses that are being reported to infect the grapevines. As the number in this pie chart says, 84 different viruses that infect grapevines. The viruses that infect both these palm fruit as well as stone fruits are very less, as you can see here. Majority of these viruses are very confined to the grapevines. that tells us something that, these viruses have specificity towards a certain host. So that brings us to the topic here, the major grapevine virus diseases. The list here does not represent the entire the list of viruses that have been reported to infect the grapevines, but it does reports that the major grapevine virus diseases in terms of their incidence or prevalence in the vineyards are listed here, starting with the grapevine red blotch disease caused by grapevine red blotch virus, and the grapevine leaf roll disease, which is a complex caused by viruses that are associated with the disease, grapevine leaf roll associated virus 1, 3, 4, and 7, and grapevine leaf roll associated virus 2. And then we have grapevine fan leaf degeneration complex, which is, again, the major causative agent of this complex is the grapevine fan leaf virus. And we do have a relatively newly discovered virus called grapevine Pinot gris virus, and also rugose wood complex, which is again associated with a number of viruses, starting with the grapevine rubus tempering associated virus, GVA, GVB, and GVT. We'll be talking about these viruses, how they can cause virus infections and how they look, especially the foliar symptoms due to these viruses in the next few slides. So starting with the red blotch, again, first thing that I would say when we talk about the symptomology, especially the foliar symptoms of different grapevine virus diseases, these can be very confusing, especially when it comes to the nutritional deficiencies. As you can see here, these are the vines, the next few slides, the images that I'm showing that we have confirmed with our molecular test in the lab, and we did measure that these are probably associated with that particular viruses or virus diseases. So as you can see, the Chardonnay leaves here, which are infected with the grapevine red blotch virus, these viruses, these symptoms can be typically deceiving when you have mineral deficiency, especially like boron deficiency or potassium deficiency. As you can see here, the yellowing symptoms on the leaves here turning into necrosis at the end of the growing season, especially. One other thing that I would point out is when you are looking for the indications of virus symptoms is to look the symptoms at the time. The timing is very important, especially virus or virus-like symptoms tend to appear more towards the end of the season after the variation. So, this particular grapevine red blotch virus, I have a video also to show you how exactly it looks like. Because of the difficulties with the volume, I'm going to play it and probably talk about it and mute the actual video volume. So bear with me a couple of minutes here, couple of seconds here. As you can see, this is a young Cabernet Franc. The video is taken on August 5th in the growing season. As you can see, these are the early symptoms on the mature leaves at the bottom portion of the canopy. As you can see, the reddening leaves symptoms here, and also on this particular leaf here, intervenal reddening, as you can see here. They look like blotchy symptoms. And these symptoms, as I said, are very limited to the bottom portion of the canopy. But as you go on and see the upper portion of the canopy, the leaves looks asymptomatic or looks even healthier. But as I said, these symptoms starts at the mature leaves and eventually progress into the upper canopy. And most of the time, we don't see those symptoms on the top portion of the canopy, even at the end of the season. Okay, so we'll go to the next one. So these symptoms on associated with the red blotch virus on the leaves of hybrid red wine grape variety that is Baconor. As you can see, they are completely different from the Cab Franc. These symptoms can be yellowing or having a necrosis at the ends of the leaf, and then eventually this necrosis also seen in the middle portion of the canopy, middle portion of the leaves. This is very particular symptoms we have observed on the hybrid grape cultivars in the last growing season, especially due to the grape vine red blotch virus. When it comes to the leaf roll on white viniferas and the Chardonnay, you can see the rolling of the leaves, which is pretty evident at certain point of the time. Again, it depends on the virus load, the virus in which type of virus that is infected, but typically the rolling of the leaves is observed on white and red fruited cultivars. On red fruited cultivars, as you can see, the Merlot leaves here, due to the grapevine leaf roll virus, the rolling of leaf edges is pretty common. Another image showing, you can also observe sometimes from the distance, especially at the end of the growing season. But again, as I said, this can be very deceiving, especially between the symptoms of, associated with the leaf roll and red blotch. As you can see in this image, one particular leaf shows blotchy symptoms, and one leaf here showing leaf roll type symptoms, which is very typical with the green leaves and the interveinal reddening. But when we tested this particular wine, it is only tested positive for grapevine leaf roll virus. That being said, we do observe like nutritional deficiency, like magnesium deficiency as shown here. And also these symptoms can be pretty confusing at times due to other pathogens, especially the crown gall, which is caused by a bacterium, Agrobacterium vitis. As you can see, the foliar symptoms of the Agrobacterium, the crown gall, is here with the red leaves, especially starting from the edges of the leaves. To be more clear on these crown gall foliar symptoms, I have a couple of videos here to show you both early symptoms as well as the late symptoms. Let me first play the first one, and again, reducing the volume here. As you can see here, this young wine, again, this particular video was taken in August. The symptoms were very clear here, and you can see the gall formation at the graft union that is very evident. And also, if you observe little keenly here, especially at this image here, the shoot that is coming from the rootstock does not show any type of these symptoms, but the actual the canopy that is growing from the cyan material having this crown gall foliar symptoms. That is pretty very common, commonly observed observation, especially when related to crown gall. And the same type of crown gall was also observed here in this second video. As you can see, the entire the foliage has turned into red color, and when we go down on the bark, as you can see, that the gall formation is very clear here. So that's one of the first things that we see. If we see the red foliage, if there is any crown gall associated with that, you can peel little bark and see if there is any gall formation out there. That is very clear indication whether you have crown gall infection or it is associated with some other virus-like infections. Okay, let's go to the next one. So that brings to the next virus that we have is the grapevine Pinot gris virus. Pinot Gris is relatively newly discovered virus, the first reported in 2012 from Italy, and then it has been reported in many grape-growing countries, both North America as well as other parts of the world. The foliar symptoms are very clear in this symptomatic strain of grapevine Pinot Gris virus, as you can see here on this image. So what we know so far about Pinot Gris virus, as I mentioned before, it has been reported in different parts of the world, including Canada. And the virus belongs to a trichovirus in the family Beta-Flexiviridae. And it is known to occur both red and white grape cultivars, as well as being reported in the table grapes. One particular aspect is very interesting when we talk about this virus is it happens to have two different strains. One is one that shows symptom with what we call symptomatic strain, and one that doesn't show any symptom stays latent in many infected vines. That is called asymptomatic strain. So far, what we have done with collaborations from Dr. Jose Ramon from Summerland Research, AFC Summerland, as well as Dr. Wendy McFadden-Smith here in Ontario, as well as we have collaboration with Deb Moreau at Kentville in Nova Scotia, and Mohamedou Fall at AFC Quebec. We have been studying this particular virus, especially in terms of its genomic variability between the symptomatic and asymptomatic strain. And I'm going to present soon on CGC and webinar on this particular topic. So I just want to say a couple of interesting points so far that what we have learned about this virus is in the samples that we have studied, both from British Columbia, Nova Scotia, here in Ontario, most of the samples remains asymptomatic, and we have found very, very rare symptomatic strains, which is obviously a good thing because the effects of negative impacts of the symptomatic strain could be very damaging in terms of wine growth and its production. So we are trying to identify what are the genomic variabilities among these symptomatic and asymptomatic strains of the grapevine Pinot Gris virus. And also another interesting aspect of this particular virus is the vector, the insect vector that could be transmitting this virus. There are reports that Erinium mites can transmit this virus, but that being said, there are more studies has to be conducted on a larger scale to confirm the possibility of the Erinium mite for the widespread of this particular virus. And also there are some other alternative hosts that have been reported, which is very widely present in Canadian viticulture growing areas like lamb quarters or white Campbell. It's been reported they could act as a potential alternative host for the Pinot Gris virus. So that brings us to the next virus disease, that is grapevine fan leaf disease. It is again worldwide in distribution, and it is soil-borne disease, which means that it can be spread by a dagger nematode, that is Ziphinema index. At least as per the reports, this particular nematode species has not been reported in Canada so far. Well, there are other type of species that are present in Canadian vineyard soils. Generally, this particular disease displays a patchy pattern in the vineyard. Again, it is due to the nematodes that are associated with the transmission. That's why you can see a patchy pattern. The infected vines especially tend to show very less growth and irregular fruit clusters. And these symptoms can be pretty confused with herbicidal damage. As you can see, these leaves here showing patchy as well as the fan-like symptoms that can be confused with kind of herbicidal damage. Again, here another image that's showing fan-like morphology of the leaf and the distortion as well as the chlorosis, and also the leaf vein yellowing can be seen in the second diagram here, and vein clearing is also one of the indications that you can find associated with the fan leaf infection. And most of the time, we also see the short internodes due to grapevine fan leaf infections when you compare to the healthy ones. So the last one in the list is the rugose wood complex. Again, rugose, as the meaning says, wrinkled or corrugated, known for its typical changes in terms of showing ridges or grooves on the woody cylinder, especially both on the scion and rootstock. This modification can vary between different cultivars, and rugose wood complex or the viruses associated with this particular disease complex can be asymptomatic in most of the European grapevine cultivars as well as the American rootstock species and hybrids, but we do have evidence that these complex can show symptoms or symptoms of venal necrosis on Vitis rupestris. As I mentioned, this is a complex. There are so many disorders associated with this. What we typically found in the literature with the names rupestris stem pitting, Kober stem grooving, or LN33 stem grooving, or corky bog. And the viruses, again, there are rupestris stem pitting associated virus as well as other VT viruses that are listed here. And one other thing that I would point out at this point is the viruses that are listed here are also known to be transmitted by the same species of mealybugs that can transmit, or soft scale insects that can transmit the grapevine leaf roll viruses, except in the case of grapevine leaf roll associated virus 2, which is not known to be transmitted by any known insect vector so far. So that brings us to the next topic here, the importance of testing. As you have witnessed in my previous slides, that not all viruses show the foliar symptoms. Some of these viruses can stay latent and does not show any typical foliar symptoms. And the symptoms can be very similar. For example, what we have seen between the leaf roll and red blotch virus, they can be similar on certain grapevine cultivars. And the testing also would indicate whether the virus, the source of the virus, whether it is contaminated or coming from the propagating material, or it is coming from any other type of virus spread. And also it will help us to take disease management options. And again, I have to reiterate here, is the sustainability of grapevine protection system depends heavily on the health status of the propagating material being free of grapevine pathogens. So let's talk about the biological and molecular virus detection methods here. So they can be broadly divided into 4 categories here. Grapevine virus diagnostics, starting with the symptom-based, as we just mentioned here. That can be non-specific, and sometimes it can be inaccurate. As we have seen, it can be pretty confusing between the nutritional deficiencies as well as the virus infections. And in terms of biological detection or biological indexing is, again, symptom-based, where we have to either take scion or rootstock material of an infected and graft them onto a healthy one and try to see if the infection can transfer from the rootstock to scion, and observe the symptoms, foliar symptoms, which can be time consuming and also labor intensive. But that's one of the strongest ways of detecting, identifying particular virus diseases. And when it comes to the serological and molecular, we do have antibody-based detection methods for majority of the viruses, but since antibodies are not available to detect all type of viruses, there is a limitation in terms of how much we can detect with ELISA-based or serological-based techniques. But when it comes to the molecular techniques, especially using PCRs or RT-PCRs, even in the PCR polymerase chain reaction-based techniques, we have both endpoint where we can say whether the virus is present or not, as well as the quantitative PCR where we can say how much virus is present. And we do have, in terms of quantitative PCR, where we can say how much virus is present, we do have two different techniques where we can say the relative concentration as well as the absolute concentration of the virus. And also we do have even high sensitivity techniques like next generation sequencing or high throughput sequencing, which we'll be talking in the next few slides. So here is the table that will differentiate between, when it comes to the molecular techniques, between a conventional PCR, real-time PCR, as well as the digital PCR. I know that this slide is spread a bit complex, but I would just highlight a couple of points here, especially in terms of the limitations with the conventional as well as the real-time PCR. The conventional PCR, as it is a size-based, and it is sophisticated, but for large scale, probably it is the best approach, especially when it comes down to the price. The real-time PCR, obviously, it is relative, as well as it depends on the reference gene when you are talking about the real-time PCR, and it is it needs a lot of optimization when it comes to the technique. When you compare the real-time PCR with the digital PCR, where you can see the absolute number, absolute titer of the virus. Again, the limitation would be the price, as well as testing on the large scale, which is time consuming, as well as it needs a lot of technical skills and knowledge. When you compare that with the high throughput sequencing, the PCRs, either it is a conventional PCR or real-time PCR or digital PCR, it is specific, but it is limited to one or two different type of viruses, which means you can't test. If you have a number of viruses to test, you need to be performing individual tests rather than one comprehensive test. Whereas the high throughput sequencing would solve that particular problem, where it is not only one test would detect any number of viruses that are present in a given sample, but also can detect if there is any new variants or viruses that are in a given sample. Obviously, there are different type of enrichment processes that are involved to target the viruses in a given sample. Either you go for a total RNA, extract the total RNA and go for a ribodepletion or a poly-A depletion, or use the total nucleic acid itself as a starting material. Or also you can use the small RNAs, which is a byproduct of virus infection in a given plant sample. And also the viral nucleic acid enrichment process, like double stranded RNA, which is one of the intermediate stage in the virus replication process. In a given sample, say for example, if a virus is a virus infected plant, you're always going to find double stranded RNAs, especially for if it is a virus that has RNA genome. And most of the viruses that infect grapes are having the RNA as its genome. And the process is pretty simple. It has once we process these samples for high throughput sequencing, the sequence reads that we get, after the quality check, we would be using those genomic sequences to assemble de novo and see whether it will matches to the host genome, like the known virus genome sequences in the genome database. And there are so many tools that are available to do that, like Virtool, as I mentioned here. So to summarize, the PCR-based techniques and high throughput sequence-based virus diagnostics. As I mentioned here, there are so many other components that are involved, especially the equipment, the time required, and the prior knowledge of the virus that is required, especially in case of PCR-based virus diagnostics. And the detection range is very critical. As I mentioned before, when you go for PCR-based diagnostics, it is narrow because we'll be doing one test at a time when you compare to the HTS, where it is comprehensive. And the suitability for large-scale applications, especially it is PCR-based techniques are more suitable at this point, but we're also working on the HTS pipelines to apply for this large-scale diagnostics. So that being said,
sorry, Sud, can I interrupt just for one moment? I apologize. Sure. We just had a question that came in, and rather than waiting until the end, I thought maybe now would be a good time to ask it. Someone's just asking for a bit of a definition on that PCR, what exactly PCR testing is. Maybe if you could just expand on what PCR testing is before moving on, that'd be great. Please and thank you.
Sure. I'll try to be more general as possible. PCR, as the definition says, polymerase chain reaction. So how does it work? So in any given sample, say like we are testing for the viruses or bacteria or any pathogen, for example. Say we need to be targeting the components in that particular pathogen. Say, for example, in terms of viruses that I'm talking about, when you look at the virus composition, there was only two components, either protein or nucleic acid. It's only two components that are there in the virus. So the proteins are mostly can be detected based on the antibodies or antigen tests like ELISA. More relative is the kind of rapid test that you are doing for COVID-19. It's an antibody-based test that targets the virus proteins. But when it comes to the PCR, we are targeting the virus genome, the nucleic acids. And especially if the nucleic acids, there are two different types of nucleic acids. One is the DNA and one is RNA. But this nucleic acid cannot be present both in a given virus. It has to be either DNA virus or RNA virus. So if it is an RNA virus, so we're not going to target the complete genome of the particular virus. We're going to target a portion of the gene, RNA, portion of the gene. So what we're trying to do in the PCR is to amplify that particular portion in a few million times so that we can detect on a gel or running through an agarose gel, for example. So if I'm clear on that, so PCR is, to do that PCR, we need the information, the genetic information, the coding of the particular gene, the sequence of that gene, ATGCs, the particular oligonucleotides. So if you have that information, so we can actually try to detect that particular virus. But if we don't, then we need to look for other options like high throughput sequencing. So hope I'm clear on that. But if you need more explanations, I can get in touch. All right. I'm going to move on to the next slide here. So the management aspect, especially the clean plant programs and the vector management. So let's start with the clean plant program, why it is important. So as these viruses, once you have virus infection in the field, for example, we cannot cure that virus. For example, if you take a bacteria or a fungal infections, you might use antifungal agents or antibacterial agents. But especially in the plants, if it is infected with the virus, we cannot cure that. So one of the best ways to manage these virus diseases is to start, if you are starting a vineyard, start with the clean vines that are free from any of these viruses or any other pathogens. So, and these viruses, the clean plant program, why it is important is when you started to start a clean plant program, the things that you need to consider is which ones are important. Obviously, as I mentioned before, there are like 84 different viruses that it can infect grapevines, but they're all important. You need to come up with a way in your geographical locations or in your particular country, which viruses that are important, that are having a negative impact on the vine growth as well as the quality of the fruit. So once you define that, the list of the viruses or virus diseases, then you can have a program designed, the clean plant program designed on the testing process of that particular pathogens. And also you need to adapt the latest diagnostic methods, because in virus detection methods or diagnostics, with the new techniques that are coming up, and also based on the research that has been done or ongoing research, whether the new viruses have been discovered, whether you need to include those ones into the clean plant program. Those are the very critical steps, especially when you're starting the clean plant certification program. And also ensure that the clean certified propagating material are available to the nurseries in larger quantities. Once the clean vines are planted, it is only half job done, but the remaining half job is to prevent the further virus infection. That's another part of the clean plant program as well. So typically, I'm giving the National Clean Plant Network in the USA an example here, because of the fact that they have been established this program from the last 20-25 years, and they do have a grapevine disease testing protocol, what they call as a 2010 protocol, where they have defined in their protocol like what are the different type of techniques they're using in their certification program, as well as the list of the viruses or pathogens that they do make sure that they're clean or free from these pathogens in the material that they're releasing into the certification program. And also recently they have approved the high throughput sequencing technique as a standard approach to make this process faster when we compare to the traditional way of how to clean a grapevine propagating material from a virus infection. And once they have tested this with the high throughput sequencing and tested negative for the list of viruses they have mentioned in the program, it is qualified for the provisional quarantine release as per the NCPN standards. As you can see here, the foundation plant services at the UC Davis have a foundation block which is registered, and make sure that they maintain this particular block with different grapevine cultivars, both cyan food stock material. They have regular monitoring and testing on those material, and they are registered in the increase block at the certified nurseries that are planted in the next stage, which again goes to the growers in the fruit production vineyards, that's the schematic way of how the certification program works in the States. That being said, in Canada, we have a different approach here, where most of the material that comes from the outside out of the country is regulated by the Canadian Food Inspection Agencies. And Canada allows the import of the grapevine material under US state certification program that are there in California, Oregon, and Washington state. And also, other than USA, CFIA allows the grapevine plant certified material from European countries, especially France and Germany are the only countries that are with CFIA approved nurseries to export the grapevine material to the Canada. But some of the material that comes in will be tested by CFIA for the regulated pathogens that are listed in the CFIA website. So that being said, excuse me. That being said, we do have a Canadian Grapevine Certification Network that is formed in 2018, and they do have both interim certification as well as the long-term certification standard. Here, I mentioned the long-term certification standard, how it works. If there is a new variety that has to be included in the certification program, first it will go for a testing process where if it tests negative for the list of viruses that the CGCN has in place for the long-term standard. I think I have that list in the next slide. Once it is tested negative for, okay, let me go back and show this list here. This is the list of viruses. I'm not going to go through all these names here, just to give you an idea of what type of viruses or pathogens that we are dealing. These are the number of viruses that are there in the CGCN long-term certification standard. And also here it says that which stage the generation 1, generation 2, generation 3, at what generation stage and which virus can be tested. As you can see, G1 covers most of these viruses to be tested. So going back again to the flow diagram here, once it is tested negative, and then it goes to the propagation process, and then which again goes to the G1A, which is again in a controlled condition, and then G2 in a more of a vineyard facility for a multiplication. And then based on the needs, it might go to the G3 and the G4 stage, where typically in a setting like an NCPN, G4 is called the certified wines that comes from the nurseries here. And if it tests positive for a certain virus, that particular material will go through the virus elimination. That includes both combination of different techniques like heat therapy or micro tissue culture, meristem tissue culture techniques, which can possibly eliminate the virus. And then again, they will be tested with the list of the viruses that are there, and as well as alternatively through the high throughput sequencing technique to make sure that they stay virus or pathogen free. Okay, this is the flow diagram that again reiterates the whole process of high throughput sequencing in the clean plant program. So as I mentioned here, in the traditional way, you will be testing for more than 30 different viruses using different techniques like ELISA or PCR. And if they test positive, again, the material will go through the virus elimination process, which can typically take from months to years, 1 year. And then in the typical traditional way, time up to 2 years. And then the retesting of pathogens, again, based on the ELISAs and PCRs, which may take time from weeks to months. When you compare that with the high throughput sequencing, which is an ongoing developing process, if it tests positive, I mean, any material that will still go to the virus elimination process, and then will be retested with high throughput sequencing or next generation sequencing. And then if it is tested negative, then probably it will go through the the propagation pipeline here, G1 to G4. I'm not going through the different steps here because of the time limitations, but just to reiterate the fact that we at Brock have initiated a large Genome Canada project, which is called Clean Plant Extraction Sequencing Diagnostics, which is the main aim of this particular project is to fine-tune high throughput sequences so that we can adapt this method for large-scale diagnostics and screening of the grapevine planting material in the clean plant pipeline. So this is again collaboration with different collaborators, partners, the CGCN, to name a few, CGCN, CFIA, and the Illumina, as well as the Genome Canada and other genomic counterparts from different provinces, as well as the University of Sherbrooke. We have a collaborative project to work on this particular project and come up with a more suitable way of high throughput sequencing, which can adapt into the clean plant program. So that being said, I would also mention we do have a project that are supported by OGWRI to develop microshoot tip culture based protocols for maintaining virus-free germplasm here at Brock, with the support from CGCN as well as the Brock. We do have initiated those protocols to maintain the germplasm repository here at Brock, and also as I mentioned, the Clean Plant program is the beginning. We do need a start clean and stay clean programs. I always recommend if you're a grower or a nursery interested in becoming certified to refer to the CGCN website, as I mentioned here, and get to know more about certification options. And when it comes to the management of grapevine virus diseases, again, reiterating the fact that the diagnostics are very critical in terms of determining the virus health status of the vines, and also the recommendations. One of the papers that are published from the Ricketts et al. In 2015 is that roguing is also plays a critical role in containing the virus infections, especially when you have insect vectors present in the vineyards, and preventing the secondary spread, especially for the leaf roll viruses, by controlling either mealybugs or soft scale insects, and closely monitoring the vineyards also helps in managing the virus diseases. Applying appropriate insecticides, especially the application timings, are also very critical to manage these insect vectors like mealybugs or soft scale insects. And also using the plant material coming from reliable sources that are really certified or tested would be very beneficial to start the vineyard with the clean virus-free plant material and make sure that those plants are free from the insect vectors as well. So with that, I would like to thank my current members of the lab and also all the collaborators. I'm not listing them anybody here, but thanks to the funders as well as all the collaborators. And with that, I would like to take if there are any questions.
Well, thank you very much for that, Sud. I think that's a fantastic presentation and very informative as well. I know a lot of industry partners and those working in and around the industry will find that incredibly informative. So we do have a couple of questions here. The first is around the ATS testing. Is that becoming the gold standard in virus detection, or is it still in the development stages to be applied as a routine diagnostic?
That's a very good question. High throughput sequencing, when it started, say like 10 years back, it's all new, and as any other techniques, it was very expensive. one other way to mention to tell that how the high throughput sequencing has changed the world is like Human Genome Project, how it started a few years back, and it was very expensive research projects at that time. But nowadays we can do the human genome probably in a matter of a few $100. So that's how the process has been developed. Of course, there are a lot of techniques that are involved, and our aim is to make sure to bring that cost to the level of regular traditional PCR-based diagnostics, especially in terms of we have options of multiplexing a number of samples in a given test, and also make sure that they are not contaminated, or make sure that the sensitivity levels remains high. So with that being said, yes, it will become a gold standard, but not at this point. But when it comes to the sensitivity, accuracy, and the comprehensiveness, I think high throughput sequencing is going to be the gold standard.
I suppose it's like so many other forms of testing, and when you look at pharmaceutical development and whatnot, it just takes time, right?
That's right.
To get to the point where the cost comes down and it becomes something that's able to be used.
Yeah. So as for any technique or any diagnostic techniques that is there that is available, the larger the amount of samples that we have, we need to test, the cost will come down basically, because it's all based on the consumables that are required.
Right. Yeah, that makes sense. Okay, one other question here for you. Is there a value to something like a G1.5 model not replicating the G1 block, but extending it in some way that doesn't amount to a commercial nursery?
So in the examples that I have shown, especially taking the consideration of National Clean Plant Network in the States, because there the volumes of the required planting material is huge when you compare to, say, like what Canada needs for in a relatively comparable way. But G1, after the G1, definitely you have the options of cutting down G2, G3, and G4 blocks, depending on the requirement. You can obviously, the purpose of G2, G3, and G4 blocks is to increase the volumes and make sure that you have enough material available for example, grafting, both rootstocks and sand material. But if you don't have the requirement of huge numbers, then yeah, definitely there is an option of either cutting down at G1, G2. That is always there. But the more number of blocks, the more generations that you go through, you also have to count the fact that the more monitoring, more testing is required, and those are the protocols that are in place with both the Canadian Grapevine Certification Network, CGCN. If you can go through that long-term certification standard, I think CGCN has taken a big initiative to include all those details, like in which generation, how much amount of testing or how much amount of monitoring is required. I think that is very important as well.
Excellent. Okay, well, I think those are the questions. And the person who asked the PCR question said thank you. That explanation was excellent. So I apologize for the interruption, but thank you for explaining that during your presentation.
Yeah, no problem. Thank you again for giving me this opportunity. It's always nice to be with the COVI lecture series and be connected with a lot of industry as well as the scientific communities. And I hope my next COVI lecture series will be in person.
I think we all feel the same way. But again, thank you so much for your time and for your presentation. As with all of these lectures, this will be available on the COVI website. It should be up there in a couple of days, so please feel free to go back on and have another watch, or share this lecture or any of the other lectures that we've done this year. And we do have, of course, the series continuing. Our next one is going to be next week. So Monday, March 7th at the same time, and it's going to be Assistant Professor in the Goodman School of Business, Joachim Schultz, along with Enology and Viticulture student Jacob Anthony Gigliotti from here at Brock, who will be doing a presentation on brand storytelling on Instagram, how wine brands create epic stories one image at a time. And having been in Professor Schultz lectures previously here in the COVI lecture series, I know that that's always an interesting discussion as well. So once again, thank you very much, Sud, for your excellent presentation. Thank you all for watching, and please join us again for another edition of the COVI lecture series. Have a great day.